This is the microbiology lecture on antimicrobial drugs. So while we're not going to dive deep into every kind of antimicrobial drug there are, we're going to hit on some important ideas and apply them to some different aspects of microbiology. A couple of important ideas. The first one is using drugs to combat disease. Okay, that's what we're doing. And that is collectively called chemotherapy. And chemotherapy in this case is not just for cancer treatment, it's using any kind of chemical to treat a disease. But most people do not often refer to it by chemotherapy. One of the goals of using drugs to treat disease is that an antimicrobial drug will act inside a host to kill the growth of microorganisms, ideally without harming the host, and that part is key. So for a drug to be effective, it needs to kill the microbe causing the infection, and it needs to not be toxic to the host. So in the environment, bacteria can do this really cool thing, and other microbes can too, called antibiosis. And what that is that when some microbes are living close to one another, they can secrete these enzymes that will stop the growth of closely related microbes around those specific microbes. So you'll see this plate, it almost looks like a disc diffusion plate with these zones of clearing around some of these colonies of microbes. So every different color, size, pattern, spot on this plate, if you will, those are called colonies, and each one that looks different is probably a different species. You can see that a few of these bacteria are very good at inhibiting the growth of other microbes around them. So this tells us that these bacteria are naturally making antibiotics that are used in the bacteria's natural environment to prevent the growth of other bacteria. So some of those we can take advantage of, some of them we can't. So an antibiotic by term is only made by other microbes. Drugs that are made in the lab are not technically antibiotics, they're called antimicrobial drugs. But even by people in pharmacy, they still call them antibiotics. Antibiotics are relatively easy to discover, but few of them have really any value for us. There are a lot of problems happening with antibiotic resistance, and those are making our need for new antibiotics and antimicrobial drugs very urgent, but it's really hard to find new drugs. More than half of the antibiotics that we use in medicine today are produced by Streptomyces bacteria, which is a bacteria that lives naturally in the soil, and it grows in these kind of long filaments. Some antibiotics that we use are produced by bacillus, and some are produced by molds like penicillin and cephalosporium. Now, you've probably heard of broad spectrum antibiotics before. The word spectrum refers to the microbial activity of a drug, the range of different microbes a drug can affect. So a broad spectrum antibiotic can affect lots of different species of bacteria. Most antibiotics and antibacterial drugs have very narrow spectrums. For example, penicillin G is really commonly prescribed for gram-positive bacterial infections. It's really only effective against gram-positive bacteria. That lipopolysaccharide outer layer of gram-negative bacteria has porins for water transport, and drugs can get through to the bacteria through those pores, but they have to be small and hydrophilic or charged and able to get through those pores. So that really limits the amount of drugs that can affect gram-negative bacteria. One of the good things about broad spectrum antibiotics is that they can target lots of different bacteria. They're often used in urgent situations before we can identify a specific pathogen. So they have great medical benefits, but they're also very likely to target the normal microbiota of the host, so your normal microbiome, right? When a patient takes antibiotics, often they get diarrhea because we're killing off the good and bad bacteria in their gut, right? Too much of that, if they have C. diff present, they can develop a C. diff infection because that's an opportunistic pathogen that can take over after a lot of the good bacteria have gone. The same thing with Candida albicans after treatment with antibiotics. It flushes out the good bacteria, creates more space, and changes the pH in the genital tract, enabling Candida albicans to grow. It's yeast, causes yeast infections. And antibiotic-resistant bacteria can flourish the same way, right? Their neighbors get killed off who aren't resistant, and then there's more room for them, and there's more nutrients and everything for them to grow because they're not being affected by the antimicrobial drug. Here's a picture showing the different spectrums of different drugs. And some of these drugs you may have heard of before, like tetracycline, acyclovir for virus infections, those sorts of things. So eukaryotic pathogens pose a very unique problem in that it's much more difficult to target a eukaryotic pathogen inside a eukaryotic host than it is to target a prokaryotic pathogen inside a eukaryotic host, because our cells are much more similar to fungi and protists than they are to bacteria. It's really easy to target those structures that only exist in prokaryotes, like their cell walls, the parts of their ribosomes, pieces of their cell metabolism. But drugs that target eukaryotic pathogens can really often be bad for the host as well. And the eukaryotic pathogen could be within the host's human cells. So in order to kill the pathogen, you have to kill those cells as well. A great example of this is viruses, right? Viruses only replicate within host cells. So in order to get rid of a viral infection, you would need to not only eliminate the free-floating viruses within the host, but also eliminate those cells that are infected with virus and pumping out more virus particles. We'll talk about viruses coming up. All right, let's talk about the action of antimicrobial drugs. Some of the key kind of points of action of antimicrobial drugs are very similar to the points of action for disinfectants and sterilization agents and things like that. So there are two different ways that antimicrobial drugs can work. The first one is being bactericidal, so directly killing microbes, right? Cidal means death. Or some antibacterial drugs can be bacteriostatic. Remember, bacteriostasis is what happens when bacteria are inhibited from growing. They're not metabolizing, they're not making proteins, but they're not dividing either. They're also not dead. If a drug can stop a bacteria from growing, making more of itself, or making toxins, proteins, if it can stop that from happening for long enough for the immune system to take over, then the patient will be able to clear the infection. So sometimes that is an okay avenue to take for antimicrobial drugs. The cell wall is a very important part of bacteria. We know that cell walls help protect bacteria from osmotic stress, right? And so damaging the cell wall can greatly weaken it and cause the cell to be much more easily lysed. We can do this with antimicrobial drugs as well. And not only can they target the cell wall itself, but they can target the synthesis of the cell wall. So if the bacteria can't make the proteins and other structures it needs for its cell wall, then it won't have a stable cell wall. So that's another avenue that antimicrobial drugs can take. A great example of inhibiting cell walls and cell wall synthesis is penicillin. Penicillin targets the teichoic acids that hold the peptidoglycan together on the gram-positive cell wall, which is why penicillin is so effective at killing gram-positive bacteria. A couple of really awesome pictures here showing you gram-positive rod-shaped bacteria before penicillin and after. Another way that antimicrobial drugs can attack a microbial pathogen is by inhibiting the synthesis of their proteins, right? So we know that if, proteins are denatured, they lose their function, right? And that's why one of the reasons that heat works so well at killing microbes. But you can also inhibit the synthesis of the protein by attacking the ribosome itself. Prokaryotic ribosomes are a little bit different than eukaryotic ribosomes, and so there are pieces of that that can be targeted by antimicrobial drugs. There's a list here that you can look at. Another great way to kill bacterial cells is to alter the plasma membrane, right? Any cell, if you injure the plasma membrane, it's going to die. Antifungal drugs like micronazole, ketoconazole, they target the sterols in the plasma membranes of fungal cells because fungal cells have different sterols in their cell membranes than animal cells do. Okay, they don't have a cell wall to target. They're eukaryotes, so we can't target specific pieces like ribosomes, but we can target sterols in the cell wall that are different between fungi and animal cells. Stopping nucleic acids from being synthesized can also inhibit a microbe, whether you're stopping DNA replication or stopping transcription and translation. We can also stop bacteria from making what we call essential metabolites. So essentially molecules that the cell needs to survive. If we can stop them from making those, then the cell won't survive. You don't need to know all the details of that. Here's a great graphic with the 5 main ways to inhibit a bacteria. All right, so let's talk about some common antimicrobial drugs. Penicillins themselves, there's a lot of them. They're all inhibitors of cell wall synthesis. There's more than 50 different related antibiotics within the penicillin umbrella. They all have that common core chemical structure that is unique to penicillin, but they have different little side chains off that common core chemical, and that's what makes the penicillins different from one another. So that teichoic acid cross-links the peptidoglycan, remember, and penicillin can prevent that. So penicillins can be all natural, made completely by other microbes, or it can be semi-synthetic, meaning that we take parts of the penicillin made by microbes and add some stuff to it in a lab to make it better. Our natural penicillins are penicillin G and penicillin V. Penicillin G has to be injected. Penicillin V can be taken as a pill because it's stable in stomach acid, whereas the other one is not. Penicillins have a relatively narrow spectrum of activity because, first of all, they're limited to gram-positive bacteria, and there are also some microbes that secrete an enzyme called penicillinase, which breaks down penicillin, as the name would imply. And so if a bacteria can break down penicillin, it can stop the penicillin from breaking it down itself. So it's like a defense mechanism. Semi-synthetic penicillins were developed because they wanted to overcome some of the barriers with natural penicillin that existed, such as resistance with the penicillinase enzyme and the narrow spectrum that it only treats gram-positive bacteria. So in order to make semi-synthetic penicillins, they get part of the penicillin molecule made by the mold, and then they add in other parts synthetically in the lab. So this gives us broader spectrum semi-synthetic penicillins, things like ampicillin and amoxicillin, but resistance to those has slowly developed over time by certain strains of bacteria. And so there are newer penicillin derivatives that have been made to try to overcome that. For example, amoxicillin with clavic acid is augmentin. You probably have heard of that one before. Some other cell wall inhibitors are here. We're not going to go into detail on them. Carbapenems, monobactins, cephalosporins. You've probably heard of cephalosporins before. So some other things you're probably familiar with, bacitracin, bacitracin, however you say it. It's a topical antibiotic. We use it for cuts and that kind of thing, first aid. It's effective against gram-positives like Staphylococcus bacteria and Streptococcus bacteria. Staphylococcus, there's lots of species that normally live on our skin, which could be bad for us if they get into a wound, so that helps. prevent infection from that. And then streptococcus, there's lots of different streptococci bacteria, and they are all pretty much bad pathogens, so it will stop infection from those as well. So really great thing to have in your first aid kit. And then we have vancomycin. So vancomycin was brought out as the first line of defense against MRSA, methicillin-resistant Staph aureus. You've probably heard of MRSA before. We'll talk more about it as we go. Essentially, every different antibiotic we've thrown at MRSA, it's evolved resistance to. But vancomycin was the next step after methicillin. And now we're seeing vancomycin-resistant bacteria as well. Then we have our antimicrobial antibiotics. Mycobacterium are those ones that don't gram stain. You have to use an acid fast stain to see them. They have that thick, waxy layer of mycolic acid around their cell walls. Things like Mycobacterium tuberculosis and Mycobacterium leprae, right? Pretty bad pathogens. So how do you treat them? We use things like isoniazid and ethambutol. Isoniazid stops the synthesis of the mycolic acid, making the bacterium much more susceptible to killing. And then ethambutol is usually given with isoniazid. It stops the mycolic acid from being built onto the cell wall properly. We can inhibit the synthesis of proteins, right? If you can stop cells from making important proteins that they need, you can kill the cells. Chloramphenicol, clindamycin, you've probably heard of clindamycin before, metronidazole. And then we have our aminoglycosides, things like streptomycin, neomycin, gentamicin, tobramycin. We don't use them very commonly, but they're there. They do have side effects, which is probably one of the reasons why they're not used very commonly, like permanent damage to the auditory nerve or kidneys. So you can actually go deaf as a result of trying to treat an infection. We have one of the first broad spectrums we're going to talk about, tetracycline. So there are a few different tetracyclines. They are produced by streptomycin or Streptomyces bacteria. It got cut off under the picture there. I apologize for that. What that does is it stops protein synthesis because it stops the tRNA from getting to the ribosome properly, and then it stops the amino acid chain from growing. So it's a good against either bacteria, gram-positive or gram-negative, really any prokaryote, and it can penetrate human tissue so that it can get to the site of the infection well. One of the barriers to producing a good medication sometimes is the ability of the medication to get to the place it needs to go to. Tetracyclines are very good at getting everywhere, really, which helps them get where they need to go. Tetracyclines will get into tissue well, but not intact cells. And because of that, that's one of the reasons they don't affect eukaryotic ribosomes. However, because it's a broad spectrum antibiotic, it will also affect your normal microbiota that you actually want to be in your body, which can cause things like Candida albicans, yeast infections, and lots of other secondary infections that come up as a result of that. They're commonly used for UTIs, some kinds of pneumonia, chlamydia, rickets. They're sometimes used for syphilis and gonorrhea. And tetracyclines were really commonly added to animal feed. It's one of the things that the FDA is trying to lessen. And then we have our macrolides like erythromycin, not so great at penetrating gram-negative cell walls, but can be good for gram-positive infections like streptococcus and staphylococcus, legionella, some kinds of pneumonia. Azithromycin, you've probably heard of before. That's another broad spectrum. There are some new drugs being developed in this category, but they have been shown to be kind of toxic, so I'm not sure how far that development is going to go. A few other protein synthesis blockers that you might not have heard of before. You don't need to know them. They're just here in case you're curious. And then remember, if we can attack the plasma membrane of a cell, we can kill the cell. We can also attack the plasma membrane by affecting the synthesis of the membrane. So polymyxin B is a great example. It's used against some gram-negative bacteria for Pseudomonas infections. It's also available in over-the-counter antiseptic ointments. And then we have our nucleic acid synthesis inhibitors. There aren't a lot of these. They aren't often used because there is a chance that they can also inhibit the nucleic acid synthesis in eukaryotic cells. A couple of exceptions to that are the rifamycins that can actually treat things like meningitis because it can penetrate into cerebrospinal fluid. Quinolones and fluoroquinolones like ciprofloxacin, Cipro, that's very good against treating anthrax. They're also used for UTIs and pneumonias, again, broad spectrum, relatively non-toxic, but resistance can develop rapidly. The other thing we can do is inhibit essential metabolites. So molecules that bacteria need to survive. If we can prevent them from getting into the bacteria or prevent the bacteria from making them, then we can kill the bacteria. And that's what sulfonamides do. So you've probably heard of sulfonamides before. It's a fairly common class of antimicrobial drugs. What they are is they're bacteriostatic rather than bactericidal. They block the synthesis of things like folic acid precursors that the cells need to survive in order to synthesize DNA and RNA and proteins. It doesn't harm us as humans because we get our folic acid from our diet or from vitamins. We don't make our own, so that's why it doesn't affect us. Bacteria make their own. All right, a little bit about antifungals. So antifungals target the sterols, like cholesterol, they're lipid molecules in cell membranes of fungal cells because fungi have different sterols than animal cells. That different sterol in fungal cell membranes is ergosterol, whereas in animal cells, we actually have cholesterol. So that difference we take advantage of with our antifungal drugs. Some antifungals that you're probably more familiar with, micronazole for cutaneous mycoses, fluconazole, which can be taken internally to treat cutaneous mycoses. If we can target beta-glucan, which is important in fungal cell walls, we can help damage the fungal cells and kill them as well. Those are used as treatment for some fungal infections. There are a couple of other kinds of drugs that are good for inhibiting fungal cells specifically. There are some other drugs that are good at inhibiting fungal cells specifically. Flucytosine stops the synthesis of RNA, and so that stops protein synthesis in fungi, but it's not used very often because it's toxic to us. A few other ones that you may have heard of here that you don't necessarily need to know about for this course. And we have to mention viruses when we talk about antimicrobial drugs because viruses are microbes, even though they're not alive, and they are very dangerous to humans. And we do have some drugs, and we're coming out with more drugs as time goes on to stop viral infections. So viruses reproduce within host cells. So it's kind of difficult to target the virus because it's in our own cells. So how do we target that without damaging host cells? So we can stop the viral particles from entering the cells in the first place. Some of the drugs we have for HIV work that way. We can stop different processes of viral replication once the virus gets into the cell. We'll get more into detail on this later, but things like uncoding, genome integration, nucleic acid synthesis. So essentially the virus making more virus inside the cell. We can try to block that. Some of the drugs for influenza can target these processes. There's also some HIV drugs that can target the virus generation processes as well. In order for virus particles to leave an infected host cell after they have been replicated multiple times, there is a process that needs to take place, and things like Tamiflu target that specific process to try to block infected cells from releasing more virus. And that will in turn give the immune system time to find those viral infected cells and kill them before they release more viral particles. We also have interferons in this category. Interferons are really interesting as antiviral drugs because they amp up the immune system, but they're also proteins that are naturally produced by our immune system. So when a cell is infected, interferons are produced, and that the purpose of those interferons is to help stop viral replication. So for some infections, we actually treat with interferon to ramp up the host immune system to attack those virally infected cells. We'll talk about protozoas going forward as well. There are some anti-protozoan drugs out there, thankfully, because some of these infections are pretty nasty. Things like malaria, there are really good drugs for malaria, but they are expensive. There's also protozoan diseases like Trichomonas vaginalis, Giardia disease, amoebic dysentery, all caused by protists that we have drugs for. We have our helminths. Helminths are multicellular animal parasites, worms, if you will. They start off microscopic, so we talk about them a little bit in microbiology. Thankfully, we have drugs to get rid of them, things like tapeworms and things like that. All right, so testing for susceptibility to antimicrobials. Essentially, we would do a disc diffusion assay. There are lots of other ways that susceptibility to different antimicrobials can be tested. This is probably the one that we're going to be most familiar with. It may not be the most widely used. There are probably better and faster ways to test for it in a medical laboratory than to take the pathogen that's causing disease, plate it, and then wait for it to grow. That takes a few days. So faster results are usually better. But if you really want to know what antibiotic best treats this specific bug, you'd look for the biggest zone of inhibition around the discs. All right, let's talk a little bit about antibiotic resistance. So resistance is becoming more and more prevalent over time to different drugs. And unfortunately, we're seeing most resistance happen with the drugs that are most commonly used, which makes sense if you think about it. So what happens during antibiotic therapy is when there is an infection and a patient starts to take antibiotics, the bacteria numbers go down, and then the patient starts feeling better, but the numbers aren't zero yet, okay? If the patient stops taking the drug too soon or takes an inadequate dose, they take less of it than they should, then we can stop the bacteria count from going down all the way. And because it doesn't go down all the way, the ones that are left that haven't died off are more resistant to the antibiotic that's being used. And because these bacteria are still around after all of the other bacteria have died, there's a lot of free room and free nutrients available for them, and they can grow very robustly, even in the presence of that antibiotic, because guess what? It didn't really affect them anyway. If an antibiotic is taken for its full course at its full strength dosage, then this black line would essentially go all the way down to zero and stay there. But if it doesn't go all the way down to zero, it's going to come back up. So when an infection is treated with antibiotics, even if it's all caused by the same species of bacteria, within that species, there will be some cells that are more likely to be killed by the antibiotic and some cells that are less likely to be killed by the antibiotic. The ones that are less likely are the ones that are going to stick around if that course of antibiotics is not sufficient. And those are the persister cells. Some of them arise from genetic mutations. Some of them can get DNA transferred to them horizontally from other cells that already have developed resistance. And once a population of cells causing an infection has gone down, then there's more room for those persister cells to come back. Different ways cells can be resistant to antibiotics, and this is kind of interesting. And this is a great summary slide for pretty much all of them. They can block the antibiotic from entering the cell, so it doesn't affect it. They can inactivate the antibiotic by producing certain enzymes that break it down. They can alter it rather than breaking it down so that it doesn't do its job. Or there are some cells that can even kind of reject that antibiotic out of the cell, kick it out of the cell, if you will, so that it can't be effective. So a lot of the problem with antibiotic resistance comes from not only improperly taking your antibiotics, but misusing them, prescribing them when they're not needed to be prescribed, so that there's more exposure of bacteria to antibiotics, which can cause more resistant strains to pop up. In third world countries, especially, we're seeing a lot of antibiotics being prescribed freely, and not even really prescribed, you can just go and get them. And when that happens, they're not taken properly, and that causes a lot of resistance as well. Also, something that's happening is that they're being overused. So if we're using antibiotics more than we need to, then they're going to lose their efficacy, because it's going to kill off all of the bacteria that would be susceptible to that antibiotic. It's going to leave all of the rest of them. So that is contributing to the rise of antibiotic resistance as well. And some really interesting statistics from the CDC about unnecessary or inappropriate prescriptions. About 30% of antibiotics prescribed for ear infections are not necessary. A lot of ear infections will go away on their own, and in fact, pediatricians are prescribing them less and less for children. Believe it or not, 100% of antibiotic prescriptions for the common cold are completely unnecessary, because antibiotics that are prescribed usually treat bacteria, and the common cold is caused by one of several kinds of viruses. So while this does happen, it doesn't help anything at all, right? You don't get an antibiotic for a cold. Even 50% of the antibiotic prescriptions for sore throats are not necessary. The only time you really need a prescription for a sore throat is if you have a strep infection. There are lots of other reasons to have sore throats that don't include a bacterial infection. There are also a lot of antibiotics being used in animal feed. Some of those are being phased out over time, and so there's already been a great reduction in antibiotics used in animal feed in the United States, but there are still more than we need. So the CDC has some great recommendations for decreasing resistance, so decreasing the rise of antibiotic resistant bacteria. Those recommendations are to finish the full regimen of antibiotics and to take the proper dosage of them. Another recommendation is not using leftover antibiotics to treat your friends or family members or yourself when infections arise. You shouldn't have antibiotics left over anyway if you took the full course, because they usually prescribe you the exact amount that you need. We should only be prescribing them when they're absolutely necessary. There's a lot of pressure on doctors from patients who don't really understand how antibiotics work and what they work on to get them to prescribe antibiotics for their patients. And so a lot of times they still do. And another important recommendation for decreasing resistance is to use the most specific antibiotic possible and to avoid broad spectrums as much as we can. And that's the end. Thank you.